disorder. So as we are now approaching the end of this workshop, if you finish, actually you are free to go. This is the last exercise, but we would like to invite you all to drop us some lines and let us know if you would be interested in learning more about the connections between structural bioinformatics and sequence analysis in a more in a different environment, not using a web server, but more using bioinformatics tools in the command line and programs installed in your own computer. So it would be a more intermediate course. Here it was mostly to get you used to these tools, but if you would be interested in knowing more and more details about the insights, about the inside of each of the algorithms, please let us know, drop us some lines, and we will be very glad to hear your opinion, what you think about this workshop.
Yeah, thank you very much, everyone, for assisting this workshop. Please, as Claudia said, please write us any doubt if you don't know, if you don't have doubts for this moment, but later or tomorrow, I don't know. You can write us and maybe we can help you. And also very important to say that if you are working with prediction of structures of disorder protein content, please really refer to the CASP and CAID in the case of disorder, because they describe the accuracy of each disorder predictor and protein modeling. So I hope that you find this course very useful. Please don't hesitate in contacting us. And yeah, you have there the GitHub to do again the exercises or obtain more information. So thank you very much to everyone.
So this...
thank you. Yeah, the recording will be available, we think, right, Claudia? They are recording.
Yeah, so we know that this is being recorded. So to our understanding, this will be available, but we are not sure how and when.
Yeah, but for sure it will be. So let's wait, everyone, to this recording. Silvia Armenta Jaime, okay. You're interested in learning how to employ the command line. Yeah, I think that many of these programs even have standalone versions. It's not only about the web tools. We decided to do an approach with the web and online tools because it's in this way it's easier. But try for the programs of your interest, and if you have any doubts, because sometimes can be tricky or something, you can write us and we will be very happy to help you if we can.
Yeah, I don't know if you have our contact information, but our email is in the GitHub page, both of our emails. So yeah, we will be very happy to help. We may plan a different workshop for a more advanced course, but yeah, in command line will be what we would like to do. We also would like to have you in person and work with you, but you know, we can't plan for that. So yeah, thanks for letting us know.
Yeah, Luis Solano is asking, do you have any specific starting points or recommendations for tools utilized in ortholog, paralog analysis? Well, Luis, in the case of the orthologs, there's a tool in the NCBI that is called COX, and it's a cluster of orthologs groups, so you can find some approach to many of the available protein sequences. But I don't know, Claudia, if you have another recommendation for tools utilized in orthologs and paralogs analysis. There are many.
Yeah, it's a hard question because I think that's the hardest part of all of this, to tell apart the ortholog and the paralogs. So do you remember the name of the tool from Pablo Vinuesa?
Yeah, get homologs.
Yeah. So there's one from that can be used. So many of the tools that tell apart orthologs from paralogs will perform a blast between query and target, and then between target and query, it will give you the best double way hit that you get. So that's one of the strategies. Otherwise, I would rely on databases because yes, telling apart orthologs, it's very hard. Another thing I would try, and this is something I've done, is there's a program in the MPI bioinformatics toolkit called GLANS, and it will make clusters of close homologs. This is not the same as finding orthologs, but at least you will get a set of proteins that are more closely related, and from this set of proteins you can tell apart orthologs from paralogs more easily. So we finished earlier than we expected. I hope you don't mind. And I think we are going to, you are welcome to hang out in here for a few more minutes. And in 10 minutes, 15 minutes, I think we are going to just close the session before it ends. But if you have anything in particular you want to discuss, you have our emails.